Hello, and welcome to this masterclass, and today we'll explore the muscular system. As a future food safety officer, understanding muscle physiology is critical. It's the bedrock of understanding meat quality and responses to foodborne pathogens. This session transforms muscles from a biological topic into a powerful tool. We're building a mental framework for your daily work. Meat and poultry are muscle tissue. Their texture, tenderness, and safety are influenced by muscle cell processes. From rigor mortis to disease signs, muscle physiology is indispensable for informed judgments. This interactive session is designed for you, aspiring food safety officers. We'll cover muscle types and contraction mechanisms to ensure you understand, not just know. This understanding separates a good officer from a great one, ensuring food supply safety. Engage, ask questions, and challenge your understanding. Let's build the foundation of your career, covering everything from actin and myosin to meat inspection. See how muscle fiber physiology impacts the food industry and public health. Let's begin and equip you for your examinations and your vital role in food safety. Let's explore the connection between muscles and food safety. The health of the animal impacts its muscle composition, affecting meat quality. Your knowledge of muscle physiology allows you to diagnose issues, linking observations to biological causes. Factors affecting meat texture and color can influence microbial growth. Recognizing subtle signs and understanding their origins is paramount as a food safety officer. The study of the muscular system is a professional necessity. It empowers you to make connections and protect consumers. Today's masterclass is structured for clear understanding. We'll begin with an overview of the muscular system, its functions, and how it works with the skeletal and nervous systems. Then, we'll dive into the three muscle types: skeletal, smooth, and cardiac, outlining their unique characteristics. Next, we'll explore the mechanism of muscle contraction and the sliding filament theory. Finally, we'll recap key takeaways and practice with multiple choice questions to solidify your knowledge. It is incredibly important that we constantly bridge the gap between theoretical knowledge and its practical application. Let's consider inspecting a poultry processing plant where chicken breasts exhibit woody breast. With the knowledge you gain today, you'll understand this condition is a metabolic myopathy. Another critical application lies in the process of aging meat. Why does hanging a beef carcass for several weeks make it more tender? Natural enzymes break down key structural proteins, tenderizing the meat. Think also about the electrical stimulation of carcasses. This process involves passing an electric current through the carcass shortly after slaughter. It accelerates the natural post-mortem processes and prevents cold shortening. Ultimately, your mastery of the muscular system will make you a more effective and discerning officer. This elevates your role, making you a true expert in the field. To succeed, rote memorization is not enough. We need a strategic approach, focusing on genuine comprehension. This masterclass builds a narrative, showing how information flows logically. We'll start with the why and then walk through the how. We focus on visual learning and simple language, cutting through complex jargon with analogies and diagrams. We repeat key concepts to cater to various learning styles. Our strategy is exam-oriented, tailored to reflect food safety officer examination requirements, emphasizing high-yield topics. Active participation is key. Engage with the material, reflect on examples, and make the concepts your own. Let's define the muscular system. It's a complex organ system of specialized cells called muscle fibers, which contract to produce force and motion. There are three types: skeletal, smooth, and cardiac, each with unique functions. The system governs everything from our heartbeat to digestion. It accounts for 40 to 50% of body mass and includes over 600 skeletal muscles. The muscular system is the body's engine, converting energy into force. It's more than just movement. It's a dynamic system crucial for health and homeostasis. For food safety officers, animal muscle mass indicates health and meat quality. It's a vital system reflecting an organism's biology. We'll explore its physiological significance and how it maintains internal stability. While the most obvious function of the muscular system is producing movement, its contributions are diverse. Locomotion and manipulation are key, with skeletal muscles attaching to bones via tendons to produce motions like walking. Smooth and cardiac muscles move substances within the body. Cardiac muscle pumps blood, while smooth muscles regulate blood pressure and enable digestion. Muscles also provide structural support and maintain posture through continuous contraction. Facial muscles enable communication through expressions, and muscles control speech. A vital function of the muscular system is thermogenesis, the generation of heat. Our bodies must maintain a constant internal temperature. The The muscular system is the primary contributor to this internal furnace. Every time a muscle contracts, energy is lost as heat, distributed by the bloodstream to maintain our core temperature. When cold, the brain triggers shivering, rapid muscle contractions to generate heat. This demonstrates the crucial role of muscles in thermal regulation. Their muscle mass acts as a metabolic engine. After slaughter, the carcass loses heat. The rate of temperature drop influences meat quality. Controlling carcass temperature post-mortem ensures a safe, high-quality product. Posture is a dynamic process, constantly adjusting our body's position to counteract gravity. This is achieved through sustained contraction of postural or anti-gravity muscles in the core, back, neck, and legs. Managed subconsciously by the nervous system, these muscles maintain balance whether sitting, standing, or walking. Good posture ensures proper skeletal weight bearing, minimizing stress on joints and the spine. Poor posture can lead to musculoskeletal problems like back pain and joint degeneration. Muscles also stabilize joints and limbs, providing a stable base for movement. Muscles play a vital role in providing dynamic stability. Ligaments offer fixed support connecting bone to bone. Muscles cross joints, providing active, adjustable support through tone and contractions. This is crucial for mobile, unstable joints like the shoulder. Rotator cuff muscles act as a dynamic cuff, stabilizing the humerus. The nervous system constantly adjusts this dynamic stabilization, using sensory feedback from proprioceptors. Reflexive muscle contractions protect joints from injury. Strong muscles are key to joint health, preventing strain on ligaments and cartilage. Muscles function as smart ligaments, adapting to loads and providing stability. This synergy allows for mobility and strength in our musculoskeletal system. Joint health is intimately tied to the function of surrounding muscles. The muscular system works with the skeletal system to form the musculoskeletal system. Bones act as levers, with joints as pivots. Muscles attach to bones via tendons, contracting to create movement like flexion or extension. Muscles often work in pairs. As one contracts, the other relaxes, allowing for controlled motion. Understanding this is crucial for food safety officers. Examining bones and muscles during carcass inspection provides insights into the animal's health and any potential injuries. This knowledge aids in making informed decisions about the carcass. The nervous system is the command and control center, directing every action of the musculoskeletal system. Muscles rely on nerve impulses to contract, ensuring precise, coordinated movement. Every movement begins as a signal in the brain or spinal cord, traveling down a motor neuron to the neuromuscular junction. This triggers the release of acetylcholine, initiating an electrical signal in the muscle fiber, leading to contraction. The nervous system fine-tunes muscle force by varying nerve signals and motor unit recruitment. Observing an animal's coordination and gait during inspection can reveal nervous system issues affecting muscle function, indicating potential food safety risks. To understand how muscles function, we need to look at the structure of a single skeletal muscle. A whole muscle is a highly organized organ composed of muscle fibers wrapped in connective tissue. These fibers are bundled together, with each bundle wrapped by connective tissue. These connective tissue layers converge to form a tendon that attaches the muscle to the bone. This structure has implications for meat quality, particularly its texture and tenderness. Muscles used for locomotion have more connective tissue and are tougher. Muscles used less frequently have less connective tissue and are more tender. The age of an animal also impacts connective tissue, with older animals generally having tougher meat. Cooking methods are often chosen to account for this. Tougher cuts benefit from slow cooking methods, which help break down collagen. Understanding muscle anatomy is key to understanding meat science and cookery. The muscular system requires substantial energy in the form of ATP. Muscles must be richly supplied with blood vessels to deliver fuel and oxygen and remove waste. These vessels form a dense network of capillaries that surrounds each muscle fiber. When a muscle becomes active, the demand for oxygen and nutrients skyrockets. Arterioles dilate, increasing blood flow. Oxygen delivered by the blood is particularly important for efficient ATP production. Muscles contain myoglobin, which can bind to oxygen and store it within the muscle fibers. The color of meat is determined by myoglobin concentration. During inspection, assess blood vessels and muscle tissue for signs of disease. Incomplete bleeding creates an environment susceptible to spoilage. Now that we understand the muscular system, let's focus on the three muscle tissue types: skeletal, smooth, and cardiac. They differ remarkably in structure, control, location, and roles. Understanding these differences is fundamental for a food safety officer. Skeletal muscle is voluntary and striated. Smooth muscle, involuntary, lines organs. Cardiac muscle, only in the heart, is striated and involuntary. Keep these distinctions in mind as we explore their anatomy and relevance to your career. Skeletal muscle is abundant in humans and animals. These muscles give the body its shape. Their primary function is to move the body. Every time you walk, run, or change your facial expression, you are engaging your skeletal muscles. The structure of skeletal muscle is designed for powerful contractions. Each skeletal muscle is a discrete organ composed of muscle tissue, connective tissue, nerves, and blood vessels. Skeletal muscle fibers themselves are remarkable cells. From a food safety perspective, skeletal muscle is the central tissue of interest. Therefore, its composition, quality, and condition are the main focus of meat inspection. Skeletal muscle is under voluntary or conscious control. When you decide to pick up a glass, a signal travels from your brain, down your spinal cord, to muscles in your arm and hand. This separates skeletal muscle from smooth and cardiac muscle, which operate autonomically. Voluntary control allows for incredible diversity and adaptability of movement. An animal that is stressed before slaughter will experience a fight or flight response. Depleted energy stores alter the pH decline in the muscle, leading to quality defects. Assess an animal's behavior and welfare, which are linked to this voluntary muscle control system. Your knowledge connects animal welfare directly to food quality and public health. Under a microscope, skeletal muscle fibers exhibit alternating light and dark bands, called striations. This pattern represents the organized internal machinery responsible for contraction. Striations are created by the arrangement of actin and myosin into sarcomeres. The dark A bands represent myosin, and the light I bands actin. The Z disc anchors actin filaments, defining the sarcomeres boundaries. During contraction, actin and myosin slide, shortening the sarcomere. This organized structure allows skeletal muscle to generate strong force. Unlike smooth muscle, striated muscle is designed for rapid movement. Striation integrity indicates muscle health and is affected by diseases and post-mortem aging. For a food safety officer, skeletal muscle is the primary commodity you are charged with protecting. A deep understanding of this tissue is the very core of your expertise. Your role requires you to assess this tissue for safety, quality, and wholesomeness. This knowledge empowers you to make informed judgments that protect consumers. One of the most common tasks you will perform is identifying abnormalities in meat, like parasitic infections or bruising. Knowing the normal anatomy and color of healthy muscle is the essential first step. Pre-slaughter stress can lead to PSE or DFD meat, which can have food safety implications. In essence, the entire field of meat science is applied skeletal muscle biology. Let's now shift our attention from the powerful and voluntary skeletal muscles to the silent, tireless workers of our internal world, the smooth muscles. This type of muscle tissue is fundamentally different from skeletal muscle in almost every aspect, from its appearance and control to its location and function. Smooth muscle forms the contractile portion of the walls of most of the body's hollow internal organs and tubes. If you imagine the vast network of plumbing and passageways within your body, your digestive tract, blood vessels, urinary bladder, respiratory airways, and reproductive tract, it is the smooth muscle that provides the motive force to control and move substances through these systems. It is the unseen, involuntary workforce that maintains homeostasis and keeps our internal environment running smoothly. Under the microscope, the difference is immediately apparent. Smooth muscle cells are small, spindle-shaped, and contain only a single, centrally located nucleus. Most importantly, they lack the highly organized sarcomere structure of skeletal and cardiac muscle. While they do contain actin and myosin filaments, these proteins are arranged in a crisscrossing, lattice-like network throughout the cell, rather than in neat, parallel rows. This is why they do not have a striped appearance, earning them the name smooth or non-striated muscle. This structural arrangement is perfectly suited to their function, which often involves squeezing and exerting pressure on the contents of a hollow organ from multiple directions. The contractions of smooth muscle are generally much slower and more sustained than the rapid, powerful contractions of skeletal muscle. Think about the slow, rhythmic waves of peristalsis that move food along your intestines. This process can take hours. This slow and steady nature is highly energy efficient, allowing smooth muscles to maintain a state of prolonged contraction, often called tone, without fatiguing. This is essential for functions like maintaining blood pressure, where the smooth muscles and artery walls must remain partially contracted continuously. This ability to sustain contractions for long periods with low energy consumption is a key feature that distinguishes smooth muscle from the more energy-demanding skeletal muscle. For a food safety officer, smooth muscle is critically important, particularly during the inspection of the viscera or the internal organs. The condition of the smooth muscle in the gastrointestinal tract, for example, can reveal a great deal about an animal's health. Signs of inflammation, abnormal contractions, or thickening of the intestinal wall can all be indicators of infectious diseases or other pathological conditions. Similarly, the inspection of blood vessels and the uterus can reveal evidence of systemic diseases. Understanding the location and normal function of smooth muscle is therefore essential for conducting a thorough and accurate post-mortem examination of the entire carcass, not just the skeletal meat. A fundamental characteristic that defines smooth muscle is its mode of control. It is entirely involuntary. You have no conscious, direct command over the contractions of the smooth muscle in your body. You cannot will your blood vessels to constrict or your intestines to speed up digestion. Instead, the activity of smooth muscle is regulated automatically by the autonomic nervous system, which is the branch of the nervous system responsible for controlling our unconscious bodily functions. This system operates in the background, constantly monitoring the body's internal state and making adjustments to smooth muscle activity to maintain a stable internal environment or homeostasis. It is a sophisticated, self-regulating system that frees up our conscious mind from the overwhelming task of managing our internal organs. The autonomic nervous system itself has two main divisions that generally have opposing effects on smooth muscle: the sympathetic and the parasympathetic divisions. The sympathetic division is often associated with the fight or flight response. When activated, it typically prepares the body for action by constricting blood vessels in the skin and digestive tract to redirect blood to skeletal muscles and dilating the pupils and airways. The parasympathetic division, on the other hand, is associated with the rest and digest state. It generally promotes functions like digestion and urination by stimulating the smooth muscles of the gut and bladder. This dual control allows for very precise regulation of organ function. In addition to the nervous system, smooth muscle activity is also heavily influenced by other factors, including hormones and local chemical conditions. For example, hormones like adrenaline can cause some smooth muscles to contract and others to relax. The hormone oxytocin stimulates powerful contractions of the smooth muscle in the uterus during childbirth. Furthermore, local factors such as changes in oxygen or carbon dioxide levels, pH, or the presence of certain chemicals can directly cause smooth muscle to contract or relax, allowing for a localized response that is independent of nervous or hormonal signals. This multifaceted regulation makes smooth muscle incredibly adaptable to the specific needs of the tissue it surrounds. This involuntary control system is a key area of focus during ante-mortem inspection. While you cannot directly see the smooth muscles contracting, you can observe the results of their function. An animal with diarrhea, for instance, is exhibiting hyperactive smooth muscle contractions in its intestines, which could be a sign of infectious or toxic conditions. An animal having difficulty breathing might have constriction of the smooth muscles in its airways. Recognizing that these outward signs are manifestations of an issue within the involuntary smooth muscle systems is a crucial diagnostic skill. It allows you to connect an observable symptom to a potential underlying pathology that could have significant food safety implications. Smooth muscle's diverse functions are best understood by examining its locations throughout the body. It's in the walls of the gastrointestinal tract, propelling food via peristalsis and facilitating digestion and absorption. In the cardiovascular system, it controls blood vessel diameter, regulating blood pressure and flow. In the urinary system, it transports urine and controls bladder function. It also plays a key role in the respiratory system and even in the eye. This underscores its fundamental importance in regulating our internal physiology. We now arrive at the third and final type of muscle, a tissue that is arguably the most impressive of all cardiac muscle. This specialized muscle is found in one place and one place only: the walls of the heart. Its very name, cardiac, is derived from the Greek word cardia, meaning heart. The function of cardiac muscle is singular and monumental to pump blood throughout the body continuously, every minute of every day, from before birth until the moment of death. This relentless rhythmic contraction and relaxation provides the force that delivers oxygen and life-sustaining nutrients to every cell in the body and carries away metabolic waste products. The function of the entire organism is utterly dependent on the ceaseless work of this incredible engine. Cardiac muscle is a fascinating blend of the features we have seen in skeletal and smooth muscle. Structurally, it shares a key characteristic with skeletal muscle. It is striated. When viewed under a microscope, cardiac muscle cells or cardiomyocytes exhibit the same pattern of alternating light and dark bands, indicating that their actin and myosin filaments are arranged into highly organized sarcomeres. This structure provides cardiac muscle with the ability to produce powerful, forceful contractions, which are necessary to eject blood from the heart and push it through the vast network of blood vessels. This shared feature speaks to a common requirement for strong directional force generation. However, in terms of its control, cardiac muscle behaves like smooth muscle. Its contractions are completely involuntary. You do not consciously command your heart to beat. It possesses its own intrinsic rhythm, generated by a specialized group of cells within the heart known as the pacemaker. This intrinsic rhythm is then modulated by the autonomic nervous system and hormones, which can speed up or slow down the heart rate to match the body's changing demands, such as during exercise or rest. This involuntary nature ensures that the vital task of blood circulation is never forgotten and continues reliably without any conscious effort on our part. For the food safety officer, the heart is a critically important organ to inspect during post-mortem examination. Its condition provides a window into the overall cardiovascular health of the animal. Pathological conditions such as pericarditis, which is inflammation of the sac around the heart, myocarditis, which is inflammation of the heart muscle itself, or valvular lesions can be indicative of systemic infectious diseases. Furthermore, signs of congestive heart failure, such as an enlarged heart, can point to chronic conditions that may affect the wholesomeness of the entire carcass. A thorough examination of this unique cardiac muscle is therefore a non-negotiable part of your inspection duties. Cardiac muscle possesses unique structural features perfectly adapted for its lifelong mission of endurance. Cardiac muscle cells, or cardiomyocytes, are shorter, thicker, and branched, forming an intricate, three-dimensional network. One of the most remarkable features of cardiac muscle is its incredible resistance to fatigue. Cardiac muscle cells are packed with mitochondria, ensuring a constant supply of ATP. Cardiac muscle has an absolute requirement for a continuous supply of oxygenated blood via the coronary arteries. The extensive sarcoplasmic reticulum and T-tubule system manages calcium ions, contributing to the long-action potential and refractory period, preventing tetanic contractions. Perhaps the most unique and defining structural feature of cardiac muscle is the presence of specialized cell junctions called intercalated discs. These are complex structures that appear as thick, dark lines under the microscope, where the ends of adjacent cardiac muscle cells connect to one another. These discs are not simply points of contact. They are highly specialized interfaces that serve two critical functions. First, they physically bind the cells together, and second, they allow for the rapid transmission of electrical signals between the cells. These functions are essential for ensuring that the billions of individual cardiac cells contract in a coordinated, wave-like fashion, allowing the heart to function as a single unified pump, a concept known as a functional syncytium. Within each intercalated disc, there are two main types of cell junctions. The first are anchoring junctions, specifically desmosomes and fascia adherens. Desmosomes act like strong spot welds or rivets, firmly holding the cells together and preventing them from pulling apart during the powerful contractions of the heart. The fascia adherens are broader anchoring sites where the actin filaments from the last sarcomere in a cell attach to the membrane, linking the contractile machinery of one cell directly to the next. This physical connection ensures that the pulling force generated by one cell is efficiently transferred to its neighbors, creating a strong, unified contractile force throughout the entire heart wall. The second, and perhaps more physiologically significant component of the intercalated discs are the gap junctions. These are essentially tiny protein channels that form a direct cytoplasmic connection between adjacent cells. These channels allow ions, and therefore electrical current, to pass directly and almost instantaneously from one cell to the next. This electrical coupling is the key to the heart's coordinated contraction. When one cardiac muscle cell is excited and generates an action potential, the impulse spreads rapidly through the gap junctions to all the connected cells, causing them to depolarize and contract almost simultaneously. This turns the entire network of individual cells into a single, electrically unified entity. This feature is what allows an electrical impulse that starts in the heart's pacemaker to spread rapidly and uniformly throughout the atria and then throughout the ventricles, producing a synchronized heartbeat. Without these gap junctions, each cell would have to be individually stimulated by a nerve, which would be an impossibly slow and inefficient system. The presence of intercalated discs is a definitive characteristic of cardiac muscle. Finding these structures in a tissue sample is a clear confirmation that you are looking at heart muscle. Their integrity is vital for normal heart function, and any disease that disrupts these junctions can lead to dangerous arrhythmias, where the coordinated pumping action of the heart is lost. To consolidate your learning and ensure you are fully prepared for your exams, let's conclude this part with a clear, concise summary of the key distinctions between the three muscle types. Mastering these comparisons is crucial, as questions requiring you to differentiate between skeletal, smooth, and cardiac muscle are very common. The three primary areas of comparison you must know are structure, including striations and cellular characteristics, control, which is whether the muscle is voluntary or involuntary, and location or function. Committing these core differences to memory will provide you with a robust framework for answering a wide range of questions accurately and confidently, really demonstrating your mastery of the subject. First, let's recap structure. Skeletal muscle is characterized by long, cylindrical, multinucleated fibers that are clearly striated. Smooth muscle consists of small, spindle-shaped cells with a single nucleus and, crucially, no striations. Cardiac muscle presents as a hybrid. Its cells are branched, typically have a single nucleus, and are striated like skeletal muscle. However, cardiac muscle has the unique defining feature of intercalated discs, which physically and electrically connect the cells. These structural differences are directly related to their different functions, from the powerful linear pulls of skeletal muscle to the squeezing action of cardiac muscle and the sustained tone of smooth muscle. Next, we review control. This is perhaps the simplest and most fundamental distinction. Skeletal muscle is the body's only voluntary muscle type, under the direct command of the somatic nervous system, allowing for conscious movement. Both smooth muscle and cardiac muscle are involuntary. Their activity is regulated automatically by the autonomic nervous system, hormones, and local factors, operating entirely outside of our conscious awareness. This distinction is absolute. If a muscle's action is something you can consciously decide to do, it is skeletal muscle. If it happens automatically, it is either smooth or cardiac muscle. Finally, location and function. Skeletal muscles are attached to the skeleton and are responsible for locomotion, posture, and heat generation. This is the meat of the carcass. Smooth muscle is found in the walls of hollow internal organs and tubes, such as the digestive tract, blood vessels, and bladder, and functions to move substances through these internal passageways and regulate pressure. Cardiac muscle is found exclusively in the wall of the heart, and its sole function is to pump blood. By remembering these three pillars, structure, control, and location, you will be able to systematically analyze any question and arrive at the correct identification and description of each muscle type. Let's explore how muscles contract to generate force. The sliding filament theory revealed that muscle protein filaments slide past one another. This causes the muscle unit to shorten. The force comes from thin actin filaments sliding over thick myosin filaments within sarcomeres. Sarcomere shortening leads to muscle fiber contraction, generating tension that moves bones. We'll now explore the molecular events driving this sliding motion. To grasp the sliding filament theory, we must understand the sarcomere, the fundamental unit of contraction. Sarcomeres are the smallest functional component of striated muscle, causing the striped appearance. A muscle fiber contains thousands of sarcomeres, forming myofibrils. Their simultaneous shortening produces muscle fiber contraction, making the sarcomere the heart of the process. Sarcomere boundaries are marked by Z discs, anchoring thin filaments of actin. Thick filaments of myosin are held in place by titin. Myosin heads grab actin, pulling them towards the sarcomere's center. This shortens the sarcomere, generating muscle power. At the molecular level, muscle contraction is a story of the interaction between two star protein players, actin and myosin. These are the contractile proteins that form the thin and thick filaments of the sarcomere, respectively. Their unique structures and their ability to bind to one another are the absolute foundation of the sliding filament theory. Let's first look at myosin, the protein that makes up the thick filaments. A single myosin molecule is composed of two intertwined heavy chains and four smaller light chains. It has a distinct shape, often described as looking like two golf clubs twisted together, with a long tail and two globular heads that protrude outwards. These myosin heads are the business end of the molecule and have two critically important features. First, they have an actin binding site, which allows them to physically attach to the thin filaments. Second, they have an ATP binding site, where they can bind and hydrolyze ATP or adenosine triphosphate, which is our body's primary energy currency. This process of breaking down ATP releases the energy that powers the entire contraction cycle. Within a thick filament, There are about 300 of these myosin molecules bundled together, with their tails pointing towards the center of the sarcomere and their heads spiraling outwards, ready to interact with the surrounding thin filaments. They are essentially the motors of the system. The thin filaments are primarily composed of the protein actin. Individual actin molecules are small and globular, known as G-actin. These G-actin subunits polymerize to form two long intertwined chains that resemble a twisted double strand of pearls. This structure is called F-actin or filamentous actin. Crucially, each individual G-actin subunit has a special active site, also known as a myosin binding site. This is the specific location where the myosin heads can attach during the contraction process. In a resting muscle, these active sites are covered, preventing the myosin heads from binding. They must be exposed for contraction to begin. The physical arrangement of these filaments within the sarcomere is what makes movement possible. The thick myosin filaments are positioned centrally, and the thin actin filaments are positioned at either end, anchored to the Z discs. The myosin heads on the thick filaments are poised to reach out, bind to the active sites on the surrounding actin filaments, and pull them towards the center. Think of it like a team of rowers, the myosin heads, in a boat, all pulling on ropes, the actin filaments, to move the boat forward. This interaction, the formation of a physical link between myosin and actin, is known as a cross-bridge, and it is the pivotal event in generating muscle force. While actin and myosin are the primary contractile proteins, they cannot initiate contraction on their own. In a resting muscle, the interaction between them is blocked by two important regulatory proteins that are also part of the thin filament complex, tropomyosin and troponin. These proteins act as the gatekeepers, controlling when actin and myosin are allowed to interact. They essentially function as a molecular switch, keeping the muscle off until the signal to contract is given. Understanding their role is crucial to understanding how a nerve impulse is translated into a mechanical force, a process we will explore shortly. Tropomyosin is a long, thread-like protein that wraps itself around the actin double helix, following its groove. In a relaxed muscle fiber, the strands of tropomyosin are positioned in such a way that they physically block or cover the myosin-binding active sites on the actin molecules. This is a simple but effective preventative measure. As long as tropomyosin is in this blocking position, the myosin heads, even if they are energized and ready, cannot latch on to the actin filaments. No cross bridges can form, and therefore the muscle remains relaxed. Tropomyosin acts like a guardrail, preventing any unwanted interaction. The position of tropomyosin is, in turn, controlled by the second regulatory protein, troponin. Troponin is a more complex protein, composed of three distinct globular subunits, that is attached to the tropomyosin strand at regular intervals. Each of these three subunits has a specific job. One subunit, troponin 1, or TNI, has a strong affinity for actin and helps to hold the troponin-tropomyosin complex in place. Another subunit, troponin T, or TNT, binds directly to tropomyosin, anchoring the troponin complex to the tropomyosin strand. The third and most critical subunit is troponin C, or TNC, which contains a binding site for calcium ions. This is where the magic happens. The entire regulatory system hinges on the presence of calcium. In a resting muscle, calcium levels within the cell's cytoplasm are kept extremely low. When the signal to contract arrives, it triggers a massive release of calcium ions into the cytoplasm. These calcium ions then bind to the troponin C subunit. This binding event causes a conformational change, or a change in the shape of the entire troponin complex. This change in shape acts like a lever, pulling on the attached tropomyosin strand and dragging it away from the myosin binding sites on the actin filament. This uncovers the active sites, effectively opening the gate and allowing the energized myosin heads to finally bind to actin and initiate the contraction cycle. Muscle contraction starts with a nerve impulse traveling down a motor neuron to the neuromuscular junction, where nerve meets muscle. At this junction, the electrical signal triggers the release of acetylcholine, which crosses the synaptic cleft and binds to receptors on the muscle fiber. This binding opens ion channels, causing sodium to rush in and depolarize the muscle membrane, generating a new action potential. This electrical signal now races along the muscle fiber, setting the stage for contraction. The action potential dives deep into the muscle fiber through T-tubules, reaching the sarcoplasmic reticulum. This triggers the release of stored calcium ions into the muscle cell. The sudden flood of calcium surrounds the myofibrils, acting as the final signal for contraction. Calcium now binds to troponin, unlocking the next phase of muscle movement. Calcium binds to troponin, causing a shape change that moves tropomyosin away from actin's binding sites. With these sites exposed, myosin heads attach to actin, forming cross bridges. This is the crucial switch. Calcium's presence turns contraction on, its absence turns it off. The system is primed for the mechanical force that powers movement. Energized myosin heads bind to actin, forming cross bridges. The myosin head releases stored energy, pivots, and pulls actin in a power stroke, generating force. After the stroke, myosin remains attached until a new ATP molecule binds, allowing detachment. The cycle is ready to repeat, driving muscle contraction. ATP is essential not just for energy, but to detach myosin from actin after each power stroke. When ATP binds, myosin releases actin, then hydrolyzes ATP to recock into a high-energy state. As long as calcium and ATP are present, myosin heads keep cycling through binding, pulling, and releasing. This ongoing process sustains muscle contraction. Each myosin power stroke moves actin only slightly, but millions of cycles create powerful, smooth contractions. Myosin heads work asynchronously, ensuring continuous force as long as calcium and ATP are available. The nervous system controls contraction strength by adjusting stimulation frequency, leading to twitches or sustained tetanus. To keep up, muscle cells rapidly regenerate ATP using creatine phosphate, glycolysis, and aerobic respiration, fueled by a steady blood supply. Just as the initiation of contraction is a precisely controlled process, so too is the process of relaxation. A muscle cannot remain contracted indefinitely. It must be able to return to its resting state to allow for controlled movement and to prevent fatigue. Relaxation begins when the nervous stimulation ceases. When the motor neuron stops firing action potentials, the release of acetylcholine at the neuromuscular junction comes to a halt. The remaining acetylcholine in the synaptic cleft is rapidly broken down by an enzyme called acetylcholinesterase, ensuring that the signal is terminated quickly and preventing any further stimulation of the muscle fiber. Without the stimulation of acetylcholine, the motor end plate repolarizes, and action potentials stop spreading across the sarcolemma and down the T-tubules. This lack of an electrical signal causes the voltage-sensitive proteins in the T-tubules to return to their original shape, which in turn causes the calcium release channels on the sarcoplasmic reticulum to close. The floodgates for calcium are now shut. With the release of calcium stopped, the dominant process becomes the active reuptake of calcium from the sarcoplasm back into the sarcoplasmic reticulum. This is an energy-demanding process carried out by powerful calcium pumps, Ki2 plus ATPase pumps, on the sarcoplasmic reticulum membrane, which use ATP to actively transport calcium against its concentration gradient. As these pumps work tirelessly, the concentration of calcium ions in the sarcoplasm rapidly decreases, falling back to its low resting level. Without sufficient calcium to bind to troponin, the calcium ions detach from the troponin C subunits. This detachment causes the troponin complex to revert back to its original shape. This, in turn, allows the long tropomyosin strands to slide back into their blocking position, once again covering the myosin-binding active sites on the actin filaments. With the binding sites now covered, the myosin heads can no longer form new cross bridges. The contraction process effectively ends. Any existing cross bridges will complete their cycle and detach as ATP becomes available, but no new ones can be formed. Without the pulling force of the cross bridges, the muscle fiber will lose its tension. It will then passively return to its original resting length, a process that can be aided by the pull of gravity or the contraction of an opposing antagonistic muscle. The sarcomeres lengthen, the I bands and H zones widen, and the muscle is once again relaxed and ready to respond to the next command from the nervous system. This entire relaxation process is just as active and vital as the contraction itself. The phenomenon of rigor mortis, which actually means stiffness of death in Latin, provides a powerful and practical illustration of the principles of muscle contraction we have just discussed, especially the crucial roles of calcium and ATP. It is a recognizable sign of death, characterized by the stiffening of the muscles of the body, which begins several hours after all vital functions have ceased. This post-mortem stiffening is a direct consequence of the chemical changes that occur within the muscle cells when the body's life-sustaining metabolic processes come to a halt. For a food safety officer, understanding rigor mortis is fundamental to the science of converting muscle into meat. After death, the circulation of blood stops, which means the muscle cells are deprived of their supply of oxygen and nutrients. The cells attempt to continue producing ATP through anaerobic pathways for a short time, but these reserves are quickly exhausted. As the cell membranes, including the sarcoplasmic reticulum, begin to lose their integrity, and the ATP-powered calcium pumps fail, calcium ions begin to leak out of the SR and into the sarcoplasm. This increase in intracellular calcium concentration triggers the same event it does in life. The calcium binds to troponin, causing tropomyosin to move and expose the actin binding sites. With the actin sites now exposed, the myosin heads, which may still have some residual energy, bind to the actin, forming cross bridges and initiating a power stroke. This causes the muscles to contract and become rigid. However, the crucial difference now is the almost complete lack of ATP. As we learned, a new molecule of ATP is absolutely required for the myosin head to detach from the actin filament. Without ATP, the cross bridges cannot be broken. The myosin heads remain locked onto the actin filaments in the rigor state, resulting in a state of continuous, irreversible muscle contraction. The entire musculature of the body becomes stiff and unmovable. This state of rigor mortis is not permanent. It typically begins 3 to 4 hours after death, reaches peak stiffness around 12 hours, and then gradually subsides over the next 48 to 72 hours. The subsequent softening of the muscles is not due to a return to a relaxed state, but rather to the decomposition of the muscle proteins themselves. Natural enzymes within the muscle, such as calpains and cathepsins, begin to break down the structure of the myofibrils, degrading the Z discs, troponin, and other proteins. This enzymatic breakdown, known as resolution of rigor, is the first step in the aging or conditioning process that tenderizes meat. Understanding the timeline and biochemistry of rigor mortis is therefore essential for managing the meat production process to ensure a safe and high-quality final product. Differentiating the 3 muscle tissue types is fundamental. Compare skeletal, smooth, and cardiac muscle based on structure, control, and location. Skeletal muscle is voluntary, striated, and moves the skeleton. Smooth muscle is involuntary, non-striated, and lines internal organs. Cardiac muscle is involuntary, striated, and found only in the heart, with unique intercalated discs. Knowing these properties is essential for identifying them during inspection. The second major area you must master is the mechanism of muscle contraction itself, which is explained by the sliding filament theory. This is a core concept in physiology and a frequent topic in examinations. The theory states that muscle contraction does not occur because the filaments themselves shorten, but because the thin filaments slide past the thick filaments, increasing their overlap and causing the sarcomere to shorten. Remember, the sarcomere is the basic contractile unit of striated muscle, extending from one Z disc to the next. The visual evidence for this theory is that during contraction, the A band, the length of the thick filament, stays the same, while the I band and H zone both shrink. At the heart of this theory are the 2 key contractile proteins, actin and myosin. You must know which is which and what they do. Myosin forms the thick filaments. Think of myosin molecules as the motors of the system. They have globular heads that can bind to actin and pivot, generating the force for movement. These heads have both an actin binding site and an ATP binding site. Actin forms the core of the thin filaments. It is a globular protein that polymerizes into long chains, and each actin molecule contains a special myosin binding site. In a resting muscle, these binding sites are covered, and this is where the regulatory proteins come into play. The regulatory proteins, troponin and tropomyosin, are the gatekeepers of contraction. This is a critical detail for exams. Tropomyosin is the long, thread-like protein that physically blocks the myosin binding sites on actin in a relaxed muscle. Its position is controlled by troponin, a 3-part complex. The key part of this complex for you to remember is its calcium binding subunit, troponin C. When calcium is present, it binds to troponin, causing it to change shape and pull tropomyosin away from the binding sites. This unlocks the actin filament, allowing the myosin heads to bind and initiate contraction. This regulatory mechanism is the molecular switch that turns contraction on and off. So, to recap, the engine room contraction is explained by the sliding filament theory, where actin filaments slide over myosin filaments. The sarcomere is the basic unit that shortens. Myosin is the thick filament motor protein with heads that bind and pull. Actin is the thin filament protein with binding sites for myosin. Critically, these sites are regulated by tropomyosin, the blocker, and troponin, the calcium-sensitive lock. Understanding the specific roles of these 4 proteins, actin, myosin, troponin, and tropomyosin, is fundamental to explaining the entire process of muscle contraction at the molecular level. The contraction of a skeletal muscle is not a spontaneous event. It is meticulously controlled by the nervous system. The entire process begins with a nerve impulse. For your exam, you must be able to trace the signal from the nerve to the muscle fiber. The starting point is the neuromuscular junction, or NMJ, which is the specialized synapse where a motor neuron communicates with a muscle fiber. When a nerve impulse arrives, it triggers the release of a specific neurotransmitter. You must know its name, acetylcholine, or ATH. This is a classic exam question. Acetylcholine is released from the neuron, crosses the synaptic cleft, and binds to receptors on the muscle fiber's membrane. The binding of acetylcholine opens ion channels, causing a rush of sodium ions into the muscle cell. This creates a new electrical signal, an action potential, in the muscle fiber itself. This is the handoff of the command. The action potential then travels across the muscle cell's surface, known as the sarcolemma, and dives deep into the cell's interior via the T-tubules. This ensures the signal reaches all the myofibrils quickly and uniformly. The arrival of the action potential at the T-tubules triggers the next critical event, the release of calcium ions. This entire sequence, from the nerve signal to calcium release, is known as excitation-contraction coupling. Calcium, or Ca2+, is the ultimate trigger for contraction. This is a point you cannot afford to forget. The sarcoplasmic reticulum, or SR, is the organelle within the muscle cell that stores these calcium ions. When the action potential arrives via the T-tubules, it signals the SR to release its stored calcium into the sarcoplasm, the fluid surrounding the contractile filaments. This sudden flood of calcium is what initiates the contraction. The calcium ions bind to troponin, which, as we just discussed, leads to the uncovering of the myosin binding sites on actin. Therefore, calcium acts as the crucial link between the electrical excitation from the nerve and the mechanical contraction of the filaments. To summarize the control system, the command starts at the neuromuscular junction with the release of the neurotransmitter acetylcholine. This generates an action potential in the muscle fiber that travels down the T-tubules. This signal triggers the release of stored calcium ions from the sarcoplasmic reticulum. Calcium is the key that unlocks contraction by binding to troponin, moving tropomyosin, and allowing the actin-myosin interaction to occur. When the nerve signal stops, calcium is actively pumped back into the SR and the muscle relaxes. This sequence, action potential, calcium release, binding to troponin, is a critical pathway to memorize. Energy is the currency of life, and in the cell that currency is adenosine triphosphate, or ATP. Muscle contraction is an incredibly energy-demanding process, and you must be able to describe the specific roles that ATP plays. It is not just one role. ATP is vital for multiple steps in the cross-bridge cycle and for relaxation. A common mistake is to think that ATP directly causes the power stroke, but this is incorrect. The power stroke is actually driven by the release of ADP and inorganic phosphate from the myosin head. ATP's roles are different, but equally critical, making this a prime area for exam questions designed to test detailed understanding. The first major role of ATP is to power the detachment of the myosin head from the actin filament. After the power stroke, the myosin head is tightly bound to actin in a low-energy rigor state. The cross-bridge can only be broken when a new molecule of ATP binds to the myosin head. This binding event changes the shape of the myosin head, reducing its affinity for actin and causing it to release its grip. Without this ATP-driven detachment, the myosin heads would remain locked to the actin filaments, and the muscle would be stuck in a state of rigid contraction. This step is essential for the cycle to continue. The second major role of ATP is to re-energize or recock the myosin head. After detaching from actin, the muscle's own enzyme activity, myosin ATPase, hydrolyzes the ATP molecule into ADP and inorganic phosphate. The energy released from this chemical breakdown is captured by the myosin head and used to reset it back into its high-energy cocked position. It is now primed and ready to bind to another actin site further down the filament to perform another power stroke. This hydrolysis step is what loads the spring of the myosin head, storing the potential energy that will be used for the next pull. The third, often forgotten critical role of ATP is powering the relaxation of the muscle. After contraction, for the muscle to relax, the calcium ions that flooded the sarcoplasm must be removed. This is accomplished by powerful calcium pumps called K2+ ATPase, located on the membrane of the sarcoplasmic reticulum. These pumps actively transport calcium from the sarcoplasm back into the SR against its concentration gradient. This is an active transport process, and it requires a constant supply of ATP to function. So, to recap ATP's three vital roles: first, detachment of myosin from actin; second, re-energizing the myosin head; and third, powering the calcium pumps to enable relaxation. The final key area of knowledge connects all these physiological principles directly to your future role as a food safety officer: the process of rigor mortis. Understanding what happens to muscle after an animal is slaughtered is fundamental to meat science and inspection. Rigor mortis, or the stiffness of death, is the stiffening of muscles that occurs several hours post-mortem. It is a direct and logical consequence of the cessation of blood flow, and therefore, the depletion of oxygen and ATP within the muscle cells. Being able to explain the biochemical basis of rigor mortis is a sign of a well-prepared candidate. The process begins as the muscle cells, deprived of oxygen, can no longer produce ATP through efficient aerobic respiration. The cell membranes, including the sarcoplasmic reticulum, begin to lose their integrity. As the ATP-powered calcium pumps fail, calcium ions leak out of the SR and flood the sarcoplasm. Just as in life, this influx of calcium binds to troponin, moves tropomyosin, and exposes the myosin binding sites on the actin filaments. The myosin heads then bind to the actin, forming the cross bridges that cause the muscles to contract and become stiff. This is the onset of rigor. The critical difference in rigor mortis is the lack of ATP for the detachment step. While cross bridges can form, there is no new ATP available to bind to the myosin heads and cause them to release their grip on the actin. As a result, the actin and myosin filaments become locked together in a permanent, rigid state. This is why the muscles become stiff and unmovable. This state is not permanent, however. The subsequent resolution of rigor is not a relaxation process, but rather the beginning of enzymatic degradation. Natural enzymes within the muscle start to break down the protein structures, particularly the Z discs, which leads to a loss of tension and the beginning of the meat tenderization process. For your exam, you should understand that rigor mortis is a normal and necessary step in the conversion of muscle to meat. The rate of its onset and the final pH of the muscle, which is affected by pre-slaughter stress and glycogen levels, are critical determinants of meat quality. Conditions like PSE, which stands for pale, soft, exudative, and DFD, meaning dark, firm, dry meat, are directly related to abnormal rigor mortis development. Therefore, a solid understanding of rigor mortis is not just abstract biology. It is applied science that is central to the duties of a food safety officer, linking animal welfare, muscle physiology, and the safety and quality of the final food product. Welcome to the multiple choice question practice and review session. Now is the time to put that knowledge to the test in a format that directly simulates what you will encounter in your food safety officer examinations. The structure of this session will be simple and interactive. I will present a multiple choice question and then reveal the correct answer with a detailed explanation. Each question has been carefully crafted to target a high-yield topic that is frequently tested and is of direct relevance to your future role. Let's begin this final, crucial step. Let's get started with our first question. All right, let's dive into our first couple of questions, which focus on the fundamental distinctions between the muscle types. Question 1. During a post-mortem inspection, you examine the wall of the small intestine. Which type of muscle tissue are you primarily observing, and what is its mode of control? The options are A. Skeletal muscle, voluntary; B. Smooth muscle, voluntary; C. Cardiac muscle, involuntary; D. Smooth muscle, involuntary. Take a moment to think. The correct answer is D. The walls of the gastrointestinal tract, including the small intestine, are composed of smooth muscle, which is responsible for the wave-like contractions of peristalsis. As we established, the function of all internal organs is managed automatically, making smooth muscle entirely involuntary. Let's break down why the other answers are incorrect. Option A is wrong because skeletal muscle is attached to bones to produce movement and is under conscious, voluntary control. You would find skeletal muscle in the main carcass, not in the intestinal wall. Option B is incorrect because while it correctly identifies the muscle type as smooth muscle, it incorrectly states that its control is voluntary. You cannot consciously will your intestines to digest food. It happens automatically. Option C is incorrect because cardiac muscle, while also involuntary, is found exclusively in the wall of the heart. This question tests your core knowledge of the location and control of all three muscle types, a cornerstone of your inspection duties. Now for question 2. A key structural feature that allows cardiac muscle to function as a coordinated unit, or a functional syncytium, is the presence of A. Multiple nuclei in each cell, B. Intercalated discs. C. A less developed sarcoplasmic reticulum. D. Non-striated fibers. Consider your answer. The correct answer is B. Intercalated discs. These specialized junctions are the unique defining characteristic of cardiac muscle. They contain gap junctions that allow electrical signals to pass rapidly between cells, ensuring a synchronized, wave-like contraction that is essential for the heart's pumping action. Without them, the heart could not beat as a single coordinated unit. Let's look at the distractors. Option A, the presence of multiple nuclei, is a feature of skeletal muscle fibers, not cardiac muscle, which typically has only 1 or 2 nuclei per cell. Option C is incorrect because cardiac muscle has a well-developed sarcoplasmic reticulum to manage the calcium needed for its powerful contractions, although it is less extensive than in skeletal muscle. Option D is incorrect because cardiac muscle is a striated muscle, sharing the striped appearance with skeletal muscle due to its organized sarcomeres. This question specifically targets the unique structural adaptations of cardiac muscle, a high-yield point for exams because it is a key differentiator from the other 2 muscle types. Let's explore muscle contraction using the sliding filament theory. During contraction, the A band remains the same, while the I band shortens. The A band represents the myosin filament, which doesn't change length. The I band contains actin filaments that slide, shortening it. The power stroke results from the release of ADP and inorganic phosphate from the myosin head. In resting muscle, tropomyosin directly blocks myosin binding sites on actin. Tropomyosin physically prevents crossbridge formation, while troponin controls tropomyosin's position. Let's focus on key molecules. Which neurotransmitter initiates muscle contraction at the neuromuscular junction? Is it acetylcholine? The answer is acetylcholine, ACA. ACA travels from the motor neuron across the synaptic cleft, triggering an electrical signal and contraction. What ion released from the sarcoplasmic reticulum triggers muscle contraction? The answer is calcium ions, Ca2+. Calcium binds to troponin, triggering the cross-bridge cycle. A new ATP molecule is required for which step in the cross-bridge cycle? The answer is the detachment of myosin from actin. ATP binding to myosin causes it to release, breaking the cross-bridge. Let's connect these principles to your work as a food safety officer with 2 questions. First, rigor mortis is primarily caused by what? The correct answer is C, the formation of cross bridges in the absence of ATP for detachment. Next, which condition in meat is associated with a rapid pH drop while the carcass is warm, often from pre-slaughter stress? The correct answer is B, PSE meat, caused by acute stress and rapid glycogen breakdown. Recognizing these defects is vital for food safety officers. We have now reached the conclusion of our masterclass on the muscular system. Throughout our journey, we've built a pyramid of knowledge, dissecting muscle types and the molecular dance of contraction. The core takeaway is the power of connection. Microscopic arrangements influence macroscopic qualities of meat. Electrochemical events at the neuromuscular junction can manifest as visible defects like PSE or DFD on the inspection line. This ability to connect the micro to the macro is the essence of being a science-led professional. Each muscle type tells a part of the animal's story, allowing you to identify signs of disease or poor welfare. This synthesized knowledge is your most powerful inspection tool. See the muscular system as an interconnected narrative from nerve impulse to food safety. The ultimate goal of this masterclass was not just to prepare you for an exam, but to empower you for a career. The knowledge you have gained today is designed to be a practical toolkit that you will carry with you into processing plants and inspection facilities. When you encounter a quality defect, such as woody breast in poultry or blood splash, ecchymosis, in a beef carcass, you will now have the physiological framework to understand the underlying cause. You will recognize that woody breast is a myopathy related to muscle growth and oxygen supply, and that blood splash is caused by a spike in blood pressure, rupturing capillaries during stunning, often before exsanguination is complete. This level of understanding elevates your role from enforcer to expert. This expertise is critical when communicating with plant managers, producers, and other stakeholders. Instead of simply stating that a carcass is unacceptable, you can explain why it is unacceptable in clear scientific terms. You can discuss how pre-slaughter handling practices directly impact glycogen levels and the subsequent pH fall, linking animal welfare to product quality. This ability to articulate the science behind the regulations builds credibility and fosters a more collaborative environment focused on prevention rather than just detection. You become a resource for the industry, helping to improve processes that enhance both food safety and animal welfare, which are inextricably linked. Furthermore, this deep knowledge enhances your critical thinking and problem-solving skills. You will inevitably encounter situations in the field that are not explicitly covered in any textbook. A novel condition, an unusual presentation of a disease, or a complex combination of factors. In these moments, it is your foundational understanding of principles, like muscle metabolism, the process of rigor mortis, and the signs of systemic disease in muscle tissue, that will allow you to reason through the problem, assess the potential risks, and make a sound, defensible judgment. It is the ability to apply first principles to novel situations that defines a truly competent and empowered officer. Ultimately, your mastery of the muscular system makes you a more effective guardian of public health. You will be better equipped to spot subtle signs of parasitic infection encysted in muscle, to identify meat that may have a shorter shelf life due to its high pH, and to understand the risks associated with improper post-mortem handling. You will be inspecting the primary protein source for millions of people, and your expertise is the final, critical checkpoint in a long and complex chain. The detailed science of muscle contraction we have studied today translates directly into safer food and a healthier public. As we bring this session to a close, I want to leave you with a final word of encouragement. The path to becoming a food safety officer is a demanding one, filled with rigorous study and significant responsibility. The sheer volume of information can sometimes feel overwhelming. However, by adopting a strategic, concept-based approach to learning, as we have done today, you can conquer even the most complex subjects. Remember that every intricate detail, from the role of troponin to the function of an intercalated disc, is a piece of a larger, logical puzzle. Focus on understanding the why behind the what, and the pieces will fall into place much more easily. Your commitment to mastering this material is a testament to your dedication to public service. The role you are preparing for is of immense importance. Food safety is a cornerstone of public health, and you will be on the front lines, ensuring that the food reaching our communities is safe, wholesome, and fit for consumption. This is a noble and vital profession. Let the importance of this mission be the fuel for your studies. When you are reviewing the steps of the cross-bridge cycle late at night, remember that you are learning the language of the very tissue you will be protecting on behalf of the public. I encourage you to continue building on the foundation we have laid today. Revisit the key points, rewatch sections that were challenging, and most importantly, keep asking questions. Engage with your peers, discuss the concepts, and try to teach them to others. It is one of the best ways to solidify your own understanding. The knowledge you build now will be the bedrock of your career, giving you the confidence and competence to face any challenge that comes your way. You have the ability and the potential to excel in your examinations, and more importantly, to become an exceptional officer. Thank you for your time and your focused attention throughout this masterclass. It has been a pleasure to guide you through the fascinating world of the muscular system. I am confident that with continued effort and a passion for the science, you will achieve your goals and make a significant contribution to the field of food safety. I wish you the very best of luck in your exams and in your future career as a guardian of our food supply. Your journey to mastery has truly begun.